Hello everybody, we are group 2 and we will talk about the observing kombucha effect on tomato plant growth and nutrition. Let's start with the introduction. What is kombucha? Kombucha, a fermented tea beverage, has grown in popularity due to its purported health advantages. Kombucha have originated in northern China around 220 BCE. This beverage is well known for its diverse treatment benefits, including antioxidant, antibacterial, anti-cancer, and anti-diabetic. It's made by fermenting sweetened tea with a symbiotic culture of bacteria and yeasts. SCOBY. The microbial community produces a cellulose biofilm at the air-liquid interface, while further metabolic activity takes place beneath the liquid surface. As the SCOBY metabolizes sugar during fermentation, organic acid levels rise and the pH of kombucha decrease, altering the tea's composition, color, flavor, and sensory properties. And the common microbiota engaged in fermentation: acetic acid bacteria, for example Acetobacter; lactic acid bacteria, for example Lactobacillus and Lactococcus; non-Saccharomyces yeast, for example Zygosaccharomyces and Saccharomyces cerevisiae. And the beneficial substances including polyphenols, vitamins, organic acids, minerals, amino acids, and hydrolytic enzymes. Excessive use of kombucha may cause health risk, gastrointestinal difficulties, or toxicity, especially for people who have mold allergies, weak immune systems, sensitive to alcohol. Kombucha is a good treatment for improving soil health by introducing beneficial microorganisms and minerals, delivering organic acids and vital nutrients, and SCOBY's mild acidity lowers the pH of alkaline soils. Now let's start with the methodology. First, 6 pots were prepared using a mixture of soil, compost, small white gravel, and 6 grams of NPK fertilizer. These materials were thoroughly mixed to ensure uniformity, and the mixture was evenly distributed into the 6 pots. Then, 2 to 3 tomato seedlings were planted in each pot. After planting, all pots were irrigated with water and placed outdoors. After 2 days, the pots were divided into 2 groups: control group and kombucha group. In control group, each pot was irrigated with 2 ml of water, and kombucha group, each pot was irrigated with 2 ml of kombucha. Here is the control group after 2 months, and here the kombucha group after 2 months. After that, the lengths of the stems and the number of leaves for each tomato plant were recorded. The roots were carefully separated from the stems and their lengths were measured. Additionally, the weights of both the stems and the roots of each tomato plant were recorded. Then, the tomato stems and root from each pot were placed in separate labeled bags. These bags were then dried in an oven for 1 day to prepare these samples for analysis. Finally, for quantifying phosphorus and potassium concentrations, ash was prepared from the dried samples and 10 ml of HCl was added to the ash. The mixture was shaken and left for 10 to 15 minutes before being filtered using filter paper into a beaker. For potassium measurement, 1 ml of the filtrate was diluted with 9 ml of water, and the potassium concentration was measured using a flame photometer. And the phosphorus measurement, 200 microliter of filtrate was pipetted into a microplate, and the phosphorus concentration was measured using the appropriate method.
And now we will start with the results. The result for the plant growth matrix generally shows that the control plants had higher average growth matrix in the case of the shoot and the root and the number of leaves compared with kombucha plants. In the case of the statistical comparison between the growth matrix for the two treatments indicates two things. The first indication, there was no significant difference in the shoot and the root length. This can suggest that the kombucha has weak effect on plant vertical development in the case of the root and the shoot. The second indication, there was a significant difference in number of the leaves between the two treatments. This can suggest that the effect of kombucha on hormone responsible for leaf production, such as cytokinase. Moving to fresh and dry weight, The result generally shows that there is relatively higher fresh and dry weight under control condition compared to kombucha treated plants, as we see in the curve. In this slide, the statistical test results indicate two things. The first indication, there is a significant difference in the shoot fresh weight between the two groups. This reduction in the shoot biomass indicates stress condition caused by kombucha, reducing water uptake through reducing turgor pressure in the shoot tissue. The second indication, there is no significant difference in the root fresh and dry weight between the two groups. This suggests that there is inhibitory effects of kombucha are more clear in the above ground tissue due to the differences in the root and the shoot hormonal sensitivity. The experiment shows that kombucha enhances root growth and increases their water retention capacity, with higher fresh and dry weights and moisture content compared to the control group. In contrast, the fresh and dry weights and moisture content of the shoot decreased, indicating a resource redistribution favoring the roots. It is suggested that bioactive compounds in the kombucha may influence hormonal balance or resource allocation within the plants. Further studies are recommended to understand the mechanism involved and to test different concentration and growth duration for optimizing its agricultural use. Moving to macronutrient quantification, potassium and phosphorus. Starting with potassium, Table 1 shows how kombucha treatments affect potassium level in the plant tissue. Potassium level were measured from the root and the shoot, and the results indicate that compared to the control group, the root and the shoot treated with kombucha has consistently lower potassium level. For example, the potassium measurement in the extract for the kombucha treated roots was 360 ppm compared to 800 ppm for the control group. The potassium measurement in the shoots was 400 ppm with kombucha and 1800 ppm with control group. In the case of dry weight, we found also that the potassium concentration in the control in the case of the root and the shoot are more than kombucha in the root and the shoot. This chart are more clearly shows that the potassium level in the control are than the kombucha in the case of the root and the shoot. So, depending on this result, we found that the kombucha treatment significantly alters potassium uptake or retention in the plant tissue. Thus, when potassium levels are low, plant processes such as photosynthesis, osmotic regulation, and its activation are affected. For phosphorus, this table shows that the phosphorus concentration in the roots and shoots increased by approximately 3 and 6-fold, respectively, compared to control group, indicating that kombucha treatment significantly increases phosphorus uptake in the plant tissue due to its microbial presence and organic acid that enhance solubility. Kombucha may enhance phosphorus bioavailability and facilitate nutrient uptake and transport. In this chart, which shows the phosphorus concentration in the plant shoots and the roots under control and kombucha treatments. In the roots, phosphorus concentration are more 3 times compared to control, and in the shoots, the phosphorus concentration are more 6 times compared to control. For the kombucha effect on the phosphorus concentration in the plant tissue, we found that the kombucha is able to enhance phosphorus absorption by plant tissue, and it contains microbes and organic acid that promote phosphorus solubilization and facilitating nutrient absorption and translocation. In conclusion, what we have learned from treating plants with kombucha is that it has two aspects, an enhancer and an inhibitor. It improves root hydration and phosphorus uptake while inhibiting shoot growth and lowering potassium level.